Hello everyone. In this video we will talk about the differences between necrosis and apoptosis. Necrosis is a set of morphological changes which is caused by denaturation of intracellular proteins and enzymatic digestion of a lethally injured cell. So what happens is when a cell gets lethally injured, it undergoes denaturation and enzymatic digestion, especially the intracellular proteins of that cell. They undergo denaturation and enzymatic digestion. The morphological changes which arise due to this process is called necrosis. Whereas in apoptosis is cell death that is induced by tightly regulated suicide programs. Suicide program means that the signal for apoptosis comes from the cell itself. It does not come from outside, or the cause is not something outside, it comes from the cell itself. So in apoptosis, the cell which is destined to die, it activates enzymes that degrades the cell's own nuclear DNA as well as nuclear and cytoplasmic proteins. In necrosis, the destructive enzymes which digest the cell, they come from the lysosomal part of the dying cell itself as well as the leukocytes. Whereas apoptosis is a genetically regulated mechanism. So, etiology of necrosis is when a cell undergoes hypoxia or toxins, which is hypoxic injury or toxic injury. That is when a cell undergoes necrosis. So it is almost always pathological. Whereas apoptosis could be physiological or pathological. It could be both. Inflammation is present in necrosis, whereas inflammation is absent in apoptosis. The number of cells involved in necrosis are multiple, because if you will see, if the cell is getting injured due to hypoxia or toxins, just one cell will not be involved. Multiple cells will be involved in that case. That is why the number of cells which are involved in necrosis are multiple. Whereas in apoptosis, a single cell is involved, because one cell's genes are involved in giving the signal for the apoptosis. In necrosis, the enzymes involved in the process are hydrolytic enzymes, which are derived from the lysosomes of the dead cell or the dying cell. Whereas in apoptosis, the enzymes involved are special enzymes called caspases. Necrosis is initiated by injury to the cell. So it could be a hypoxic injury, injury due to toxins, physical agents, chemical agents, and microbes. Whereas apoptosis is initiated by a loss of normal cell survival signals. Cell death in necrosis is by mechanisms like ATP depletion, damage to the cell membrane, and free radical injury. Whereas in apoptosis, the damage is by activation of intrinsic and extrinsic pathway, which in turn activate caspases. If we talk about morphological differences between necrosis and apoptosis, in the cell size, in necrosis, cell size is increased. The cell will show a swelling. So the size will increase in necrosis, whereas in apoptosis, the cell will show a shrinkage. Size will be reduced. Cell membrane gets disrupted in necrosis, whereas the cell membrane in apoptosis remains intact. Cell organelles in necrosis are released outside after the membrane is ruptured. So after the cell membrane gets ruptured, the cell organelles are released outside. Whereas in apoptosis, the cell organelles get packed into small apoptotic bodies. One example of which you might know is Councilman bodies, which are seen in hepatitis C. Phagocytosis in case of necrosis is done by macrophages, and these macrophages will ingest the digested cell debris. So what will happen is, Once the cells are digested by enzymes released from the lysosomes, macrophages will phagocytose these digested cell debris, remaining debris. Whereas in apoptosis, the macrophages will phagocytose apoptotic bodies, which are formed in apoptosis. If we look at the cytoplasm in necrosis, it appears vacuolated, moth-eaten. It will show myelin figures and it will be increasingly eosinophilic. So it will appear very pink under microscopy. It will show vacuolations. It has a moth-eaten appearance. Myelin figures can be seen and it will be eosinophilic. Myelin figures are phospholipid remnants. Whereas in apoptosis, in the cytoplasm, we see cytoplasmic blebs. If we look at the nucleus in necrosis, it will show karyorhexis. Karyorhexis means fragmentation. So the nucleus will appear fragmented. In pycnosis, the nucleus will be shrinked, and karyolysis means complete disappearance of the nucleus. Whereas in apoptosis, we will see a chromatin condensation. This is the characteristic feature of apoptosis.